Hello students, welcome back to my channel. In this class, we are going to discuss about one of the important topic. It is endosperm. In the previous class only, we have discussed about the endosperm while explaining about the double fertilization. Let us quickly glance about the double fertilization. In the double fertilization, we have studied that the syngamy and triple fusion. So what is syngamy? You know that one of the male gamete fuses with the egg. So after the entry of pollen tube into the embryo sac, one of the male gamete fuses with the egg, resulting in the formation of a diploid zygote. That process is called as syngamy, or can also be called as fertilization. And why zygote is diploid? Because one haploid male gamete and haploid female gamete will be fusing. That's why zygote is always a diploid structure. Then another male gamete fuses with the two polar nuclei which are present in the central cell. So this is the central cell. In the central cell we can find the presence of two polar nuclei. So another male gamete fuses with these two polar nuclei. Or else we can write in another way, because these two polar nuclei before fertilization they fuse to form one nuclei which is a diploid, and it is called as secondary nucleus. You can write the fusion of other male gamete with the two polar nuclei, or else you can write the fusion of another male gamete with the diploid secondary nucleus. Both are same. You can write two polar nuclei. If you mention two nuclei, these are called two polar nuclei. If you mention only one nuclei which is a diploid, then it is called as a secondary nucleus. So you can write the fusion of another male gamete with the diploid secondary nucleus. It is called as triple fusion. Why it is called as a triple fusion? Because here three nuclei will be fusing. You can understand here, here you can better understand, because one of the male gamete which is a haploid nuclei fuses with two haploid polar nuclei, or else here it is a diploid nuclei. With that, one of the haploid male gamete will be fusing. As three nuclei will be fusing, this process is called as triple fusion. Then what is the product of triple fusion? So after the fusion of one male gamete with the diploid secondary nucleus, it results in the formation of a triploid structure called as a primary endosperm nucleus. We can simply write it as a PEN. What is PEN? It is a primary endosperm nucleus, and this primary endosperm nucleus is the product of triple fusion. So after triple fusion, this entire central cell becomes a primary endosperm cell. The central cell becomes a primary endosperm cell which contains a nucleus called as triploid primary endosperm nucleus. So after fertilization, you know that this zygote later develops into embryo. So the stages of embryo development we will be discussing later, and this primary endosperm nucleus develops into a nutritive tissue called as endosperm. So this entire thing what you can see in the central cell, the central cell only gets converted into the endosperm, which is a nutritive tissue. Why it is called nutritive tissue? Because it will provide nourishment for the developing embryo. This embryo requires nourishment. That nourishment will be provided by this endosperm. That's why endosperm is always a nutritive tissue, provides nourishment for the developing embryo. And very importantly, you should note here the endosperm in case of angiosperms. In case of angiosperms, the endosperm is always a triploid. Then why it is a triploid? Because it is the product of triple fusion. Here primary endosperm nucleus is also triploid. At the same time, the endosperm formed from the primary endosperm nucleus also triploid in nature in case of angiosperms. But it is entirely different in case of gymnosperms. In gymnosperms, there will be a formation of endosperm, but it is not a triploid. Instead of that, it is a haploid. Try to remember. The endosperm in angiosperms is a triploid. The endosperm in gymnosperms is haploid. Why it is haploid means there is no double fertilization in case of gymnosperms. That's why the gymnosperms, endosperms are haploid in nature. So let us see how the endosperm will be formed within the embryo sac. As already I told you, after fertilization, this primary endosperm cell which contains a nucleus which is called as a primary endosperm nucleus. So this primary endosperm nucleus will divide several times, which divides repeatedly and forms a nutritive tissue called as endosperm tissue. And very importantly, so this is the endosperm tissue. Each cell of the endosperm tissue which is rich in the reserve food material. The endosperm cells are rich in reserve food materials, and that reserve food material will be utilized by the developing embryo. And depending upon the mode of formation, the endosperm is mainly classified into three types. One is nuclear endosperm. The second type is called cellular endosperm. And the third type is called helobial endosperm. I told you, depending upon the mode of its formation, endosperm is mainly of three types. One is nuclear endosperm, other one cellular endosperm, and third type is called as helobial endosperm. Let us discuss one by one in detail. So firstly, we need to discuss about the nuclear endosperm. So this nuclear endosperm, it is the most common type of endosperm development in case of flowering plants. Let us see how it will be formed. See, so this is the embryo sac which contains an embryo, and so we are discussing after fertilization. Here the zygote developed into embryo and which contains a primary endosperm nucleus. And here I have not mentioned it as an endosperm because we are discussing the formation of endosperm or development of endosperm. So this primary endosperm nucleus which undergo several or successive divisions which are free nuclear divisions. Try to understand. Here the embryo sac contains embryo as well as the primary endosperm nucleus. This primary endosperm nucleus undergo successive free nuclear divisions. They are free nuclear, that is without the cell wall formation. Without wall formation. You may be knowing that during the cell division, karyokinesis is followed by cytokinesis. What is karyokinesis? Division of nucleus. And what is cytokinesis? Division of cytoplasm followed by the cell wall formation. So here in the nuclear endosperm, in this case the primary endosperm and primary endosperm nucleus undergo several divisions which are only free nuclear divisions without the cell wall formation. So you can understand only karyokinesis will be taking place without cytokinesis. So this stage of endosperm development is called as free nuclear endosperm development. And these free nuclei will not remain as it is. Later, each nuclei will be surrounded by a cell wall. So finally, this free nuclear endosperm becomes a cellular endosperm. Try to understand. Here primary endosperm nucleus undergo division. Several successive divisions will be taking place and all are free nuclear divisions only without the cell wall formation. So resulting in the formation of large number of free nuclei. You can see a large number of free nuclei will be formed, but they do not remain as it is. In this stage we can say it is a nuclear endosperm. But each nuclei later subsequently it is surrounded by a cell wall, and finally the free nuclear endosperm becomes a cellular endosperm. So this is a free nuclear endosperm. Later it gets converted into the cellular endosperm, that is in case of nuclear type. And the second type is called as cellular endosperm. So in this case, so this is the first case. So in the second case, cellular endosperm. Here each and every divisions of the primary endosperm nucleus are immediately followed by cytokinesis. So each and every time the primary endosperm nucleus divides, which immediately followed by the cytokinesis. For example, understand here it is the primary endosperm nucleus which divides to form two nuclei. Initially, karyokinesis occurs resulting in the formation of two nuclei, which immediately followed by the cell wall formation. So here we can find two cells now. Two cells. Later, these two nuclei will divide to form two more nuclei again, which is followed by the cell wall formation. Even here also this nuclei will divide to form two nuclei followed by cell wall formation. Like that, several divisions will be taking place, and all divisions includes both karyokinesis and cytokinesis. Whenever the nucleus divides, immediately it is followed by the cytokinesis. That's why this cellular endosperm. The endosperm is cellular from the beginning only. Here at the end of this stage we can find cellular endosperm, but in the cellular endosperm from the beginning only the endosperm is cellular, because each and every division of the primary endosperm nucleus is followed by the cytokinesis. That's why it results in the formation of cellular endosperm. I can give you one best example how this free nuclear and cellular endosperm can be seen. So already I told you this embryo and endosperms will be present within the seed, and you know that in the previous class we have studied about the ovule. Ovule after fertilization develops into seed. So within the seed we can find the presence of embryo as well as endosperm. One best example, the coconut water. The coconut water in the tender coconut, which is very familiar to you. I think all of you are familiar with the coconut water which is present in the tender coconut. So what the coconut water you will consume, that is nothing but a free nuclear endosperm which is rich in thousands of free nuclei. The coconut water is nothing but free nuclear endosperm with thousands of free nuclei. And what the white colored kernel part you are eating, that is nothing but cellular endosperm. So in a coconut seed we can find both cellular endosperm as well as nuclear endosperm. The coconut water is nothing but the free nuclear endosperm, and white kernel part you are eating, that is cellular endosperm. Now we will discuss the third type of endosperm, that is helobial type of endosperm. So this helobial type of endosperm can be seen only in the order Helobiales. When you study the systems of classification, if you study the Bentham and Hooker classification, under the monocotyledons, there is one order called as Helobius, which contains a special type of endosperm called as helobial type of endosperm. Then how this will be formed? See, this helobial type of endosperm, it is intermediate between the nuclear as well as cellular type of endosperm. It is an intermediate. So both are taking place simultaneously. Let me explain. So this is the embryo sac, which contains embryo as well as a primary endosperm nucleus. So initially at the beginning, this primary endosperm nucleus will divide to form two nuclei. So initially at the first division, this primary endosperm nucleus divides to form two nuclei, which immediately followed by the cell wall formation, but resulting in the formation of two unequal cells. Try to remember, here there will be a karyokinesis resulting in the formation of two nuclei, and these two nuclei will be surrounded by a cell wall resulting in the formation of two unequal cells, the larger towards the micropylar region and the smaller towards the chalazal region. So when you study the ovule structure, we can understand the micropyle and chalazal end that I have explained in the previous classes only. So here the karyokinesis is followed by cytokinesis, but resulting in the formation of two unequal cells, the larger micropylar cell and the smaller chalazal cell. Later, so next divisions will be free nuclear type. Here the cell wall formation occurs, means this is a cellular type now. Here cell wall formation occurred, but later the next divisions are only free nuclear type of divisions. In this case, both in the smaller cell as well as in the larger cell, only the nucleus will be dividing. So it results in the multinucleate stage of the embryo. multinuclear stage. Here they are not surrounded by cell wall. So here we cannot say it as a cellular endosperm. Here it is a free nuclear endosperm because it contains a multinucleate condition. But later, again, the next divisions will be followed by cytokinesis. Each and every nucleus will be surrounded by the cell wall. So finally, this aerobial endosperm also becomes cellular endosperm because the nucleus will be surrounded by the cell wall formation. So finally, it becomes a cellular endosperm. So most commonly we can observe the cellular type of endosperm, but the development is usually most common is nuclear type of endosperm. But even this nuclear endosperm also later develops into a cellular endosperm. So these are the three important types of endosperm. Then what is the importance of endosperm? What happened to the endosperm during the development of embryo? So when you observe the structure of ovule, within the ovule, around the embryo sac, there will be a nutritive tissue called as nucellus. You may be knowing this in the anatropous ovule. So the embryo sac is present within the nucellus. So this nucellus, during the growth of this endosperm, now this will be the endosperm. Let us imagine here. So around the embryo sac, you can find the presence of nucellus. So during the growth of endosperm, this endosperm will crush the nucellus. It will crush the nucellus, and in turn this endosperm will be eaten by the embryo. Because embryo requires nourishment, that nourishment will be obtained from the endosperm. So this endosperm will be eaten by the embryo. But this endosperm may be completely utilized by this embryo, or sometimes it may remain or it may persist. When you observe the seeds, certain seeds contain a part of the endosperm which is called as a persistent endosperm. In some other plant, some other seeds, this endosperm will be completely used up. If the endosperm remains in the seed of certain plants, such a seed is called as endospermic seed, or it can also be called as albuminous seeds. You may be studied in your first PUC. If the seed contains endosperm, or we can say that if endosperm is persist or remains in seeds of certain plants, such seeds are called as endospermic seeds or albuminous seeds. For example, we can give castor, cereals, and coconut. Already I have given example, coconut, which contain both nuclear as well as cellular endosperm, which is nothing but the seed contains endosperm. It is not completely used up. Then when the endosperm will be completely used at the time of seed germination. When you sow the seeds will start to germinate. During that time, the remaining endosperm will be completely used up by the seeds at the time of germination. And in some other cases, this endosperm is completely utilized by the developing embryo. Before the seed formation only, the endosperm will be completely used up by the embryo. Such type of seeds are called as non-endospermic. Non-endospermic are also called as X-albuminous seeds. Here albuminous is nothing but they contain endosperm. X-albuminous is nothing but they do not contain endosperm. For this, I can give you example: pea, beans, sunflower, etc. So these are the plants in which the seeds do not contain endosperm, and these are the plants in which the seeds contain endosperm, and we will study these types of seeds later in this chapter only. So that completes endosperm. Endosperm is very, very important. It is a nutritive tissue because it provides nourishment for the developing embryo, and the types of endosperms are nuclear, cellular, and helobial, and even the importance of the endosperm is also very, very important. That completes endosperm. In the next class, we are going to discuss about the embryo. Here, during the development of endosperm, even the embryo also differs. The structural changes can be observed in the embryo also, but here I have not specified. While explaining about the embryo, you will able to know how the structure will be changing in the embryo. So in this class, we will be starting with the embryo.